In this section, I'm going to provide an introduction to the field of virology. So early virus discoveries. Louis Pasteur, developed a vaccine for rabies, and he originally coined the term virus, which is Latin for poison. A couple other guys discovered that the tobacco disease was caused by a virus. So tobacco mosaic virus causes mosaic-like patterns to destroy tobacco leaves. And although tobacco isn't too popular today, way back when it was a major cash crop. So when this virus was wiping out large fields of tobacco, it was a huge issue. Then a couple other guys discovered that foot and mouth disease was caused by a virus as well. The filterable virus. So infectious fluids were passed through porcelain filters that were designed to trap bacteria. The cell-free filtered fluid remained infectious, which proved that an agent smaller than a bacterium was the cause of disease. Viruses can infect every type of cell, including bacteria, algae, fungi, protozoa, plants, and animals. Different types of viruses can infect those cells specifically. There's very few viruses that can infect every cell type or multiple cell types. Usually they're very specific in what they can infect. Because they can infect every cell type, they have played a major role in the evolution of all organisms on Earth. So some unique properties of viruses. They are infectious particles. So we don't call them organisms, we call them infectious particles. They're either active or inactive. We don't call them alive or dead. So if they're inside of a host, they're considered active. If they're outside of a host, they're considered inactive. They're obligate intracellular parasites. Obligate. If you have an obligation to do something, you have to do it. So obligate means it has to. Intracellular is inside of a cell. Intracellular, inside of a cell. Parasites. A parasite is an organism relationship where one benefits and the other is harmed. So obligate intracellular parasites. They cannot multiply unless they invade a specific host cell. They must instruct the genetic and metabolic machinery of the host cell to make and release new viruses. So when a virus gets into a cell, it tells that cell to turn into a virus-producing factory. And that virus-producing factory will stop its normal everyday activity and switch to just producing viruses. That ultimately ends in that cell's death. So some properties of viruses that you should be familiar with. Viruses are not cells. They're obligate intracellular parasites of all organisms on Earth. They do not independently fulfill the characteristics of life, because one of those characteristics is you have to be able to reproduce, and viruses can't do that without a host. They are inactive macromolecules outside of the host cell and active only inside of host cells. They have a basic structure of a protein shell, a capsid, surrounding a nucleic acid core. They are ubiquitous. Ubiquitous means that they can be found anywhere, and they've had a major impact on the development of biological life. They're also ultra microscopic, which means they're smaller than what you can typically see with a normal bright field microscope. So they're 20 nanometers to 1,000 nanometers in diameter, and you typically need an electron microscope in order to visualize viruses. They can have either DNA or RNA, but not both. They can have double-stranded DNA, single-stranded DNA, single-stranded RNA, or double-stranded RNA. The RNA can also be positive and negative sense, which is something that I'll talk about later on. They can carry molecules on their surface that determine the specificity for attachment to the host cell. Those molecules, typically called spikes, allow that virus to stick to and infect a very specific type of cell. They multiply by taking control of host cells' genetic material and regulating the synthesis and assembly of new viruses. They lack enzymes for most metabolic processes, so they can't really do their own metabolism. And they lack the machinery for synthesizing proteins, so they can't make their own proteins. They need the host cell in order to do that for them. Now I keep saying the word host. If you were to host a party, you're the person who is having all the guests come to your house or come to a particular location, and you're kind of in charge of everything. So when I'm talking about a host cell, I'm talking about the cell that gets infected by the virus. So it's hosting the virus, it's going to produce more viral copies, and it's going to release those viral copies to infect other host cells. The viral size range is smaller than the average bacterium. Electron microscopes are required to detect them. Parvoviruses are 0.02 micrometers in diameter. Mimiviruses are 450 nanometers in length, and that's larger than some small bacteria. And then cylindrical viruses are 0.8 micrometers long, but 0.015 micrometers in diameter. So here you can see the relative range. So this is a yeast cell, this is a yeast cell bud, this is E. coli, just your typical bacterium. Staph, this is about the size of that. And then it kind of goes down from there. So rickettsia is a pretty small bacterial cell. And then this mimivirus is the largest known virus that exists, all the way down to the yellow fever virus, which is about the size of a molecule of hemoglobin. Now, if you think about a red blood cell and hemoglobin, there's about 250 molecules of hemoglobin in a red blood cell. So a little bit less than that of yellow fever viruses could fit in one of your red blood cells. And your red blood cell is the smallest cell in the body, or one of the smallest cells in the body. So it's pretty crazy when you think of how many viral particles can infect or can replicate inside of one individual cell. You can have a lot of viral replication before that host cell ends up rupturing. Viruses do not look like cells. This is an electron micrograph picture of viruses. They lack protein synthesizing machinery, and they need only the parts required to invade and control the host. So they have an external coating and a core containing nucleic acids. So I'm going to skip ahead to just show you this picture real quick. So when I go through the definitions of everything, you can kind of visualize this as I'm talking. So here's the capsid. The capsid is just the outer coat, and here you can see the capsid of an enveloped virus. We'll talk about the difference between a naked virus and an enveloped virus in just a bit. So this capsid contains genetic material, and that's the nucleic acid. Nucleic acid can be DNA or RNA. So there's DNA viruses and there's RNA viruses. Same thing over here. Here's your capsid, and then here's your genetic material. Viruses have spikes, and they're these protrusions that are very specific, and they bind to receptors on cells. Every virus has a receptor that it's going to bind its spike to in order to infect a cell. If the spike doesn't match the receptor on the surface of the cell, it will not be able to bind to it, and it won't be able to enter into the cell. So if you think about you getting the common cold, your dog is not going to contract the common cold from you, because the receptors in us that the rhinovirus were to bind to is very different from any receptor inside of the dog, and the rhinovirus cannot bind to those dog receptors. So the exact same type of virus could not infect human cells and dog cells. Now there are of course exceptions to this, and there's some viruses that have a broad host range. Those viruses have mutated to have less specific receptors, and their interaction between the receptors are less specific. A good example is rabies. You know how many animals and even people can get rabies. Another example would be the coronavirus, COVID-19. So that can infect numerous different types of species. We know that bats and tigers, cats and dogs and humans have all been infected by COVID-19. So there are some exceptions to this, but typically it's a very specific recognition between the spike and the host cell receptor. And that's what determines what cells can be infected by this virus. Additionally, you can see here that there's another layer around this one that's missing from this one. So that's the difference between a naked and an enveloped virus. A naked virus is just this capsid with the genetic material on the inside and the spikes. An enveloped virus has an envelope. This envelope is made up of the cell membrane of the host cell. So if you think about it, this virus, when it's getting into your cell, this membrane will kind of unfold and fuse with your host cell membrane. And when the virus is replicated and it's about to leave your cells, it rips off a chunk of your host cell membrane and takes it with it. So we'll talk more about this envelope and how viruses infect cells more specifically a little bit later. But that's the main difference, is enveloped virus has its envelope. It's going to be made up of the host cell membrane, while naked viruses do not. So going back to the definitions here, a capsid is the shell that surrounds the nucleic acid. Combined together, the nucleocapsid is the capsid and the nucleic acid. The envelope is not found in all viruses. It's usually a modified piece of the host cell membrane. And then spikes are found on both naked and enveloped viruses. They project from either the nucleocapsid or the envelope, and they allow viruses to dock with their host cells. So again, enveloped viruses have an envelope. Naked viruses consist only of the nucleocapsid. And a virion is a fully formed virus able to establish infection in a host. So a virion is typically singular. One virion is able to infect a cell. And here's a stained fluorescent image of some viruses. So here you can see the capsid, and here you can see the DNA core, the capsid, and the envelope. And these are labeled with antibodies and exposed to fluorescent light to be able to see the detail here. So the genome is talking about the full complement of DNA and RNA that's carried by a cell. Viruses contain either DNA or RNA, but not both. The number of viral genes is small compared to that of a cell, and they possess only the genes necessary to invade host cells and redirect their activity. Viruses can have a wide variety of different types of nucleic acids, so they can have double-stranded DNA, single-stranded DNA, double-stranded RNA, and single-stranded RNA. And of the single-stranded RNA, it can be positive sense or negative sense. Positive sense single-stranded RNA genomes are ready for immediate translation into protein. So when that gets into the host cell can read it right away and make a protein from it. Negative sense RNA need to convert it back into DNA, and then they can convert it into proteins. So they kind of go backwards and then forwards with that. So positive sense is ready to go straight from RNA to protein, while negative sense has to go back to DNA, and then it can go forward to RNA. So here are some examples of DNA viruses and RNA viruses. You do not need to know which ones are which. This is just, if you're curious, an example of all the different types of genetic material that you'd find inside of, inside of the different viruses. Other substances that you can find inside the virus particle include certain enzymes. Polymerases synthesize DNA and RNA. Replicases can copy RNA. Reverse transcriptase can synthesize DNA from RNA. And then there's substances from the host cells. So arenaviruses pack along host ribosomes with them. Retroviruses borrow the host's tRNA molecules.